Hello, class. So this is the third and final part of the series, Biosynthesis of Amino Acids. And so for this talk, we will be discussing feedback inhibition regulates amino acid biosynthesis, and amino acids are precursors of many biomolecules. And so, as far as the regulation of amino acid biosynthesis, there is something that regulates it that's called feedback inhibition. And basically, that is taking the actual product, the final product, it actually inhibits the commitment step of the enzyme that is used to catalyze and make your initial reactant into the product. So you don't have as much product anymore. So that is what is called feedback inhibition. And so an example of feedback inhibition is serine biosynthesis. And so serine is the final product, and it actually inhibits the enzyme called 3-phosphoglycerate dehydrogenase, which is the enzyme that converts the 3-phosphoglycerate into the serine. And so this reaction should look familiar because this was shown in the last part 2 of this series, where, remember, you have the NAD plus, which oxidizes and forms this double bond, and then you have glutamate that transfer a free amino group and also releases the alpha keto glutarate, and then you have that phosphoryl group that's replaced with hydroxyl group via hydrolysis. And so a representation of this enzyme is pictured here, where the enzyme which catalyzed the committed step in the serine biosynthetic pathway includes a serine binding regulatory domain, which is right here. Serine binding to this domain reduces the activity of the enzyme, which is that feedback inhibition. And so there's also a combination of feedback inhibition and activation that helps regulate amino acids. And so this example here, you have hydroxyethylethylamine pyrophosphate. And when it is reacting with the alpha ketoglutarate, it ends up forming this isoleucine. And this isoleucine is actually inhibits the threonine deaminase. And this is the enzyme that converts threonine into alpha ketobutyrate, which ultimately ends up as the final product of the final product being isoleucine. So that's how you have that feedback inhibition, because this is the final product, and so it is actually inhibiting the commitment step, where that conversion of threonine is being converted down to isoleucine. So now you're not going to have as much isoleucine when it is inhibiting the enzyme that is used to create this amino acid. But also, if this hydroxyethylethylamine pyrophosphate goes a different route and does a reaction with pyruvate, you end up making leucine and valine, and the valine actually activates this threonine deaminase. So valine activates, which is not part of the actual product from the breakdown of threonine. It's something outside, and so that helps activate it. But what is the threonine final product of isoleucine? Once again, it causes a feedback inhibition And so it blocks the activity of the threonine deaminase. And so just like the phosphoglycerate dehydrogenase having an active site for serine that blocks um, the commitment step of the enzyme, the threonine deaminase also has an binding amino acid binding site for the isoleucine to help deregulate or stop the commitment step of the threonine deaminase. And so another type of feedback inhibition is cumulative feedback inhibition. In cumulative inhibition, each inhibitor can reduce the activity of the enzyme even when other inhibitors are bound at saturating levels. The enzymatic activity of glutamine synthetase is switched off almost completely when all final products are bound to the enzyme. And so this is showing that example of glutamine synthetase, which is the enzyme that converts glutamate into glutamide. This should look familiar. This was discussed in the first part of this series, where you have glutamate. It goes through an ATP-dependent reaction where it attaches the phosphoryl group, and that phosphoryl group gets replaced with ammonium ion, and then you have a free amino group to have your glutamine. And so this glutamine would act as a feedback inhibitor, but also glutamine is a source of nitrogen in the synthesis of these other products that are listed. And so since it is part of creating these products, all of them would inhibit this glutamine synthetase. And that's, since it's not just one, that's what makes it a cumulative feedback inhibition. Okay, and so the activity of glutamine synthetase is also controlled by reversible covalent modification, um, modification of the attachment of an AMP, which is listed right here. So this unit by a phosphodiester bond to the hydroxyl group of a specific tyrosine residue in each subunit. So this is the tyrosine residue. You have the phosphodiester bond right here, and this that's highlighted in pink is your AMP. And so this structure actually inhibits the glutamine synthetase as well. And so as far as glutamine synthetase activity and it being the activity being less active versus more active. So this adenylated enzyme is less active and more susceptible to cumulative feedback inhibition than is the adenylated form. The covalently attached AMP that we discussed before unit is um, is removed from the adenylated enzyme by phosphorol, phosphorol, phosphorolysis. Phosphorolysis. And so that shows right here where um, you have your phosphorolysis. That means that phosphoryl group is being added. And so from there, this enzyme, the adenylated glutamine synthase is more activated. But when it is uh, the when you have the AMP being added to um, the unit, and that's what this reaction is for, and releases that pyrophosphate. So now that phosphorus group that was attached is now taken up. Now you have this regulatory protein, that's what's called PA. Um, it means it's more, it is activated, that protein is activated to make that enzyme less activated. And so that's why it has a PA and a PD. When this protein, which acts as an inhibitor, when that protein is activated, it will officially inhibit the enzyme adenylated glutamine synthetase. But when that protein is not activated, then you have a more activated, which is called the adenylated glutamine synthetase. And this notate that AT that's shown here stands for adenylyl transferase. So not that typical ATP that you would see in before. So this don't confuse the two. And so with this regulatory protein, where PA means that it is activated and actively inhibits the enzyme, it actually does a transition from a activated form to a deactivated form by uridine monophosphate. And so you have UTP that donates that uridine monophosphate and then ends up deactivating the regulatory protein. But if you have ATP alpha ketoglutarate, that activates this movement to have it deactivated. And so when it's deactivated, that means it is not inhibiting that main source of enzyme that is creating glutamine. But glutamine is the final product. Thus, if there's more glutamine around, it will try to stop that regulatory protein from being converted to deactivated form because it want it to be activated. That's why it can slow down the formation of glutamine. So PA it shows green as in it's activated and it's trying to slow things down for the enzyme, thus stopping that glutamine being produced. But as far as if you have a deactivated enzyme and you want it to become activated, then you need to have glutamine that will activate it. So these plus and minus symbols minus means inhibits, and so deactivate. That plus symbol is activation. So it's driving it to go into from a deactivated form to an activated form. And once again, just trying to hold this in. When it's in this activated form, it is trying to slow down or stop the enzyme that produces glutamine. And so as far as another product that is not part of the products of glutamine, which is the alpha ketoglutarate, if more of that is available, it's going to try to stop it from going into the activated form because it's going to be like, oh, hey, we want to produce more glutamine, and so it needs to stay deactivated. Now, the uridine monophosphate is actually released from the regulatory protein by hydrolysis. And finally, as far as this amino acid derivatives, we talked about a lot of different amino acids that act as precursors for many molecules. These are just some additional ones just to keep in mind as far as your general education, but we already went through some of the major molecules that are created from amino acids, and that includes the series for the biosynthesis of amino acids.